Hello everyone. Today I will be showcasing a project where I use a machine learning algorithm to predict the venom type of different snake species based solely off the protein composition of their venom. So for context, I'm currently working on a project pertaining to snake venom evolution, and I have this dataset. This dataset contains 16 columns. The first column represents the names of all the species I have in my dataset, and the last column represents the type of venom that each snake species produces, either neurotoxic or hemotoxic. To provide background, neurotoxic venom comprises toxins which affect the victim's nervous system, whereas hemotoxic venom comprises toxins which affect the victim's cardiovascular system. Now, the 14 columns I have in the middle here each represent different proteins that are within the venom of each species. These are represented as proportions. For instance, the first column represents the different proportions of the venom protein PLA2. And for the first snake species, you can see that this protein takes up 71.2% of its venom composition. I will be using this dataset to train a machine learning algorithm in Python in order to create a model that can predict the venom type of a snake species after feeding it only the venom protein compositions. But before that, let's visualize this data. In this file, I've written a script to present the dataset in what is known as a PCA plot. So if we run this file, this is what we get. Each point on this graph represents a snake species that you saw in the dataset, and each point is either colored blue if the species has neurotoxic venom, or red if the species has hemotoxic venom. Ultimately, this PCA plot converts all the correlations among all the venom proteins into a two-dimensional graph. So, points that are more highly correlated tend to cluster together. This is why blue points tend to cluster more on one side and red points tend to cluster more on the other. So how can we use this data to create a model that can predict venom type just based off of venom protein proportions? For instance, if a new data point was added to this PCA plot, like the yellow one I added on the graph here, it might be simple for a person to say that this point probably represents a neurotoxic species, or a blue point, because it tends to cluster around other blue points. But how would a machine learning algorithm know whether to classify it as neurotoxic, a blue point, or hemotoxic, a red point? In this project, I will be employing an algorithm called support vector machine to do just that. A support vector machine creates what is known as a hyperplane in such a way that all points of one category are on one side of the plane and all points of another category are on the other side of the plane. So any new point added, a support vector machine can classify it into one category or the other based off what side of the plane the point lands on. To create this plane, a support vector machine model uses a set of training data. In this case, the training data will be this dataset which I showed before, where each species is already labeled as neurotoxic or hemotoxic. Now, before we use the whole dataset to train our model, this first file I created instead splits the dataset into random training and testing subsets. Here is a representation of these subsets. This split is done so that we can both train our model and later evaluate it to see how accurate it is. In this case, approximately 70% of our dataset will be used to train the model and 30% will be used to test the model. However, remember that the testing subset fed into the model will not contain the corresponding venom types. The model will try to predict these venom types solely based off the input venom protein proportions. Afterward, we will compare the predictions to the actual venom type values to assess the model's accuracy. Now, when I run this Python script, a support vector machine algorithm will be fed my training subset. Once trained, the program will then be fed my testing subset and make corresponding venom type predictions. So, running this code will output the results from the testing subset of the data, where we can see how many of the model's venom type predictions match the actual venom type labels. So let's run it. As shown on the screen, we get the results from our testing data of 23 different snake species, which is approximately 30% of my entire dataset. As you can see, in this run, the model got 20 venom type predictions correct and 3 venom type predictions incorrect. The model even outputs the true neuro rate, which is the proportion of actual neurotoxic species that were correctly identified by the model as neurotoxic, and the true hemo rate, which is the proportion of actual hemotoxic species that were correctly identified by the model as hemotoxic. Note that if we run the model again, Because the training and testing subsets are randomized each time, the model may output slightly different results, but by and large, the outputs will be similar. Finally, to visualize the utility of this machine learning model more clearly, I've created a last Python file which trains a support vector machine using the entire dataset here, not just a subset. After training the model, I will feed the model this other incomplete dataset. The model will make Venom-type predictions based off of this. Notice, in this dataset, there are no Venom-type labels for any of these 35 species of snakes. There is only the input values of protein proportions. So, the model will take this dataset and predict the Venom types of all 35 species. Afterward, to assess the accuracy of the model's predictions, I will compare them to the Venom-type values I have noted down for each snake species, which I have obtained from various scientific sources online. So, if we first run the model and train it using the original complete dataset, then we input the dataset without the labeled Venom types of which the model will predict, we get the following output. Now, as you can see, the model's predictions are in the image on the left. On the right is a table containing the actual Venom types, sourced from various scientific journal articles, which are in the rightmost column of the table. The model predicted the Venom type of 33 out of 35 snake species correctly, thus it is fairly accurate. Overall, this model can be used to accurately predict the Venom types of snake species, after being fed solely the protein component inputs. And, as snake venom continues to be of great importance for biomedical research, more datasets will continue to be created with more venom protein composition data, which can be used to further train this model, increasing its overall accuracy.